So hi everyone, welcome back to the last introductory lecture. And this lecture is going to put together the concepts we learned in the first two lectures on biology and technology. And we'll learn about the experiments that were performed to generate the data for the data science challenge and how we're going to work with that data during the challenge. So the data itself comes from an experiment of Perturb-seq done in T cells. And so let's start talking about the experimental overview, and then after we understand the experiment, we'll go into what the data looks like. So the experimental overview. So first, I just want to start with a recap from what we learned in the first two introductory lectures. So the first lecture, we learned that T cells become exhausted in the cancer environment, and they transition from progenitor T cell states to effector T cell states, and finally terminally exhausted T cell states. These transitions are driven by the gene expression programs in the T cells, and the T cell states are marked by different abilities to kill tumor cells, with the effector T cells and the progenitor T cells being much better at killing tumor cells or cancer cells than the terminally exhausted T cells. So that's a recap of what we've already learned in the first lecture. And in the second lecture, we introduced Perturb-seq, which allows us to do a simultaneous readout of the gene expression in a single cell, as well as the perturbation that cell received. And how that worked is we used the CRISPR-Cas9 gene editing technology to knock out individual genes in the T cells in parallel. And then we introduced those T cells into cancer, let the T cells fight the cancer for some time, isolated the T cells, and then used sequencing to read out both the perturbation that that cell received, as well as the resulting gene expression in that single cell. And so this lets us read out perturbations in parallel, as well as the gene expression measurements or expression values for single cells. Okay, so with those two recaps, the T cell exhaustion and how Perturb-seq works, we can now talk about the experiment that was performed by Mark Schwartz, who is a physician scientist and oncologist at the Broad Institute and Dana-Farber Cancer Institute. And so what experiment was performed was Mark took mice who had melanoma, and he gave them T cells. And these T cells will migrate into the tumor, into the melanoma, and they will fight the cancer. And 2 weeks later, Mark took out the tumor and isolated the T cells and performed single-cell RNA sequencing, which we saw in the second lecture. And what he saw when analyzing those T cell states that came out of the single-cell RNA sequencing analysis was that a high proportion of T cells were in the terminally exhausted T cell state. And that's not surprising because we know that in the tumor environment, T cells get driven towards terminal exhaustion. Now, then Mark wanted to know, if we take these mice with melanoma, what happens if instead we give them T cells that have been genetically edited? And now individual genes are knocked out in these T cells, and we can ask, do these gene edits, when we knock out a gene, does this affect the ability of the T cell to respond and kill the cancer cells, to kill the melanoma? So Mark prepared T cells with different genetic perturbations. He gave those to the mice with melanoma. Those T cells proceeded to fight the melanoma, and they went through their differentiation pathways. And then after 2 weeks, Mark isolated the T cells from the tumor and perform Perturb-seq. And the outcome of the Perturb-seq experiment is we have single cells, and for every single cell, we know what genetic perturbation that cell received, as well as the resulting gene expression in that same single cell. And so when we analyze the gene expression for those single cells, we might find that perturbation A was associated with T cell killing, so the effector T cell state, and perturbation B might have been associated with the exhausted T cell state. So that's the experiment that was done, and this is what you're going to be working with in the Data Science Challenge. So let's just go through a little bit of question and answer with some more details of this experiment. So first question you might have is how many genes were knocked out? So remember, there are 20,000 protein coding genes in the human and mouse genomes. So how many genes were actually knocked out? Well, Mark knocked out 73 of those genes, and this is, not 20,000, because as we said in the previous lecture, the amount of work to do that, it would take about 50 years. So we did a more manageable number of genes. And then most genes are going to be in the training set, and 7 have been left out for validation and testing. And this is what you're going to be working with in the first challenge, those 7 genes, and predicting their effects on the T cell states. Next question is, well, why did Mark choose those specific genes? So We don't know what most genes do or what most genes, how most genes affect T cell differentiation. So Mark used prior knowledge to kind of narrow the search space. So one thing he did was a scientific kind of literature review of genes with potential roles in controlling differentiation. Mark also looked at human patient data from melanoma trials. And he looked at genes from T cells in melanoma, in particular T cells that were expressing TCF7, which is a critical gene in T cell differentiation. And he looked at what genes mark those T cells. So that's like where he chose the genes from. And then we can ask, how many single cells were sequenced? So we did this Perturb-seq experiment, and we isolated T cells. How many cells did Mark isolate? So Mark isolated 70,000 cells. And because there's 70 genes, that means there's about 1,000 single cells for each genetic perturbation on average. And then finally, are there negative controls in this experiment? Are there perturbations that are not expected to have an effect? And actually, yes, there are. The experiment was carefully designed to have negative controls. So there are 30 what are called non-targeting controls, which are perturbations that are not actually going to make any cuts in the genome and affect any gene in the cell. And these just have no effect on the gene expression, no effect on the T cell states. And so this can give us a background sense of what unperturbed T cells would look like in the context of this melanoma model. So the data that comes out of the Perturb-seq experiment consists of two pieces. The first is the gene expression matrix, and the second is this assignment of the perturbations that each cell received. So remember, the gene expression matrix will tell us the expression level of every single gene in every single cell. Now, this perturbation table tells us what perturbation, what genetic perturbation a cell received. So cell 1 received a tox perturbation, so the tox gene got deleted. And then we can link that perturbation to the resulting effect on the gene expression within that cell. So cell 1 had a tox deletion and it has this set of gene expression values. So that's this really nice linkage we can do in a Perturb-seq experiment between the perturbation and the result on the gene expression in the same cell.